Figure 3.15, Multiple Memory System Model. According to Squire and Zola Morgan, 1991, different anatomical regions of the mammalian brain are associated with different subtypes of memory. Okay, so it's a diagram of the brain with different parts labeled. You can see the striatum, procedural memory, the prefrontal cortex, working memory, the amygdala, classical conditioning of emotional responses, hippocampus and medial lobe, declarative memory, neocortex, perceptual priming, and the cerebellum, classical conditioning of simple reflex responses. If this model of memory function is true, it raises the question of why evolution would favor the emergence of qualitatively distinct memory systems rather than a single system that could adjust to different environmental constraints. One possibility is that specialization arose in memory processing because cognitive systems that store one type of information are better at solving particular cognitive problems. For example, episodic memory may have evolved when humans started living in groups and had to keep track of who did what to whom. Memory for these personal events is unlikely to facilitate memory for a motor skill. Indeed, cognitive processes supporting procedural and episodic memory may be incompatible in that skill-based learning depends on extrapolating commonalities across repeated occurrences of an action, while ignoring the what, where, and when of each incident. Episodic memory is the reverse, defined by information tagged to specific events, rather than generalities across events. This distinction would produce a functional incompatibility that is the basis of the multiple memory system model. Sherry and Schrachter, 1987. Finally, a memory system that appears to serve more than one function may arise if a system that evolved to serve one function has been co-opted by another. Neuroscience of memory. In the multiple memory systems model, the acquisition, storage, and retrieval of different types of information are associated with distinct neural substrates. But what happens within these systems? Are there general properties of memory processing across systems? Although the pieces of the puzzle are not yet complete, current evidence points to commonalities in the cellular, molecular, and synaptic mechanisms that underlie different types of memory. Cellular mechanisms. Most of what we know regarding the cellular basis of memory comes from the Nobel Prize-winning work of Eric Kandel and his colleagues using the marine sea snail, Aplysia californica. The animal has a relatively simple nervous system, making it a prime model for studying neural circuits that control behavior, as well as how these are altered during learning. A common behavioral measure in these studies is a reflex withdrawal response when the animal is touched. Aplysia exhibit both habituation and sensitization of this reflex. As expected, habituation develops to a mild stimulus, whereas sensitization develops to a stronger one. Over habituation trials, sensory neurons release less of the excitatory neurotransmitter glutamate, causing a reduction in the firing of motor neurons controlling the response. Figure 3.16. These changes occur at several sites in the reflex circuit, suggesting that memory is not confined to a specific location in the circuit. The same synapses that are depressed during habituation are enhanced during sensitization. In this case, the circuit is modulated by an interneuron that releases increasing amounts of serotonin, 5-hydroxyltryptamine, 5-HT, with each stimulation. 5-HT activates sensory neurons, causing intracellular changes that make it easier for the neuron to fire in the future. This potentiated response in sensory neurons increases the firing of motor neurons and, by consequence, the magnitude of the reflex withdrawal response. The same process has been described in other invertebrates and in some vertebrates, suggesting a common mechanism for reflex modification across species. Figure 3.16, Cellular mechanisms of sensitization in aplysia. A strong, potentially aversive touch to the aplysia body, e.g. the tail, introduces a withdrawal response, mediated by glutamate release from the sensory to motor neurons. If the sensory stimulation is repeated, withdrawal responses become stronger, sensitization. This is mediated by increased release of serotonin, 5-HT, from interneurons connecting sensory and motor neurons. Okay, so there's a diagram of these aplasia. They have a mantle, gill, head, periodum, tail, and siphon. And in the second diagram, it shows that they have sensory neurons, facilitating interneurons, 5-HT, glutamate, and that's next to the gill. And then they have motor neurons, sensory neurons, siphon skin, a tail, sensory neurons, and the mantle shelf. Oh, okay, so they have sensory neurons all around them. Changes in neurotransmitter release are short-term, so they cannot explain long-term changes in habituation or sensitization that aplasia or other animals display. With sensitization, repeated 5-HT release causes intracellular changes that activate genes, thereby producing new proteins. These proteins alter neuronal function and lead to structural changes, such as the growth of new synaptic connections. In contrast, long-term habituation leads to a loss of synaptic connections, although the mechanism that produces these changes is not as well understood. The structural changes associated with sensitization involve a long cascade of molecular changes, one of the most important being activation of the protein ch3. Research on aplasia, as well as the common fruit fly, Drosophila melanogaster, implicates ch3 function in very simple forms of learning. Genetic and pharmacological manipulations in rodents demonstrate that ch3 is also required for more complex forms of memory, including spatial and social learning. In addition, some human disorders characterized by severe deficits in learning, such as Alzheimer's disease and Coffin-Lowry syndrome, are associated with altered ch3 function. Thus, molecular mechanisms of memory may be conserved across species with ch3-mediated gene expression, acting as a universal modulator of processes required for memory formation. Keep in mind, however, that there is no single genetic marker for memory. Even apparently simple forms of learning recruit multiple genes and proteins which act synergistically to modulate neuronal function and communication. These intracellular changes may span an extended time frame, with some phases occurring immediately and others up to 24 hours following an experience. 5-HT is not the only neurotransmitter to be implicated in memory. Decreased acetylcholine levels in Alzheimer's disease have linked this neurotransmitter to memory, although acetylcholine-related deficits in these patients may reflect a disruption in attentional processes. This raises an important point. Given that memory has a role in most cognitive function, it is often difficult to determine whether a particular biological substrate has a role only in memory or in memory as it interacts with other cognitive processes. Synaptic mechanisms. The molecular and cellular changes described in the previous section change the way that neurons communicate with each other. The Spanish neuroanatomist and Nobel laureate Santiago Ramón y Cajal was one of the first to suggest that memory involves improved neuronal communication, but more than a half-century passed before the idea caught on. Most people attribute this hypothesis to Donald Hebb, 1949, and his cell assembly theory. With no experimental evidence and no way to test it, Hebb proposed that connections between two neurons are strengthened if they fire at the same time. As a corollary, it becomes easier for one neuron to excite the other in the future, i.e., for a presynaptic neuron to cause an action potential in a postsynaptic neuron. According to Hebb, this mechanism of enhanced synaptic connectivity is the physiological basis of memory. It was close to 25 years before Hebb's theory was confirmed experimentally using brain slices extracted from a rabbit hippocampus. Bliss and Lomo, 1973. In this preparation, which has now been used in thousands of studies, see Figure 3.17, Electrodes are placed in the bundle of axons entering the hippocampus and in the group of neurons to which these connect. First, the axons are stimulated with a single electrical pulse, and action potentials are recorded in the postsynaptic neurons. This provides a baseline measure of synaptic strength. Second, the axons are stimulated with repeated bursts of high-frequency pulses. Third, a single pulse is applied to the axons, and postsynaptic responses are recorded. Postsynaptic responses are enhanced in the third phase, compared to the first phase of the experiment, and the effect can last for weeks. This persistent increase in synaptic strength following high-frequency stimulation of presynaptic neurons is called long-term potentiation, LTP. Long-term potentiation results from increased responsivity of postsynaptic neurons, not increased neurotransmitter release from presynaptic neurons. Interestingly, when trains of pulses are presented at a slow rate, the strength of the connection is decreased, a phenomenon known as long-term depression, LTD. Figure 3.17, long-term potentiation, LTP, in the hippocampus. Responses of a group of neurons in the CA1 region of the hippocampus are recorded when two inputs are stimulated alternately. Following this baseline testing, a string of high-frequency stimulation is applied to input 1. Responses are then measured to stimulation of each input. LTP is manifested as an increased response to stimulation of input 1, but not input 2, following the high-frequency stimulation. Okay, so we can see a diagram. It's showing the CA1 neuron and a recording electrode touching it, and then we're seeing high-frequency stimulation to input 1 and no stimulation to input 2. Okay, then we have a couple of graphs. So on the x-axis of the first one is time in minutes from 0 to 40 minutes, and on the y-axis is response to stimulation of input 1 with no numbers. Okay, so at the beginning you can see that the baseline responses go about a quarter of the way up the y-axis at the beginning, and then by 30 minutes in, they're doing the second thing, and you can see that the response is much higher. It's about all the way up the y-axis, showing the LTP, the long-term potentiation in the hippocampus, because a burst of high-frequency electrical impulses has been sent. In the second graph, the x-axis is still time in minutes from 0 to 40 minutes, and the y-axis is now response to stimulation of input 2. Okay, so then the stimulation is added in at 20 minutes, and you can see that the baseline responses carry across all the way across the graph about a quarter of the way up, so nothing has changed. So maybe this is showing that they're only stimulating the first neuron, not the second one. Long-term potentiation has a number of properties that make it a plausible neurobiological substrate of memory. For one, it is synapse-specific. Postsynaptic neurons may have hundreds of inputs, but only those that are stimulated with high-frequency pulses are enhanced at a later time. Additionally, inputs will become associated if they are activated together. A weak input that does not elicit an action potential in postsynaptic neurons will acquire this ability if it is stimulated with a strong input. These two properties relate to memory in that organisms remember only a small amount of specific information from previous experiences, and two events that occur together may become associated. Classical conditioning is one of the best examples. Further evidence for a long-term potentiation memory connection is that manipulations which block long-term potentiation also block learning, and long-term potentiation is mediated by a cascade of intracellular changes similar to those that mediate memory, e.g. protein synthesis. Perhaps the strongest evidence that long-term potentiation is a cellular form of memory comes from an occlusion study, Whitlock et al., 2006. In this experiment, memory for an aversive event induced changes in brain chemistry that mimic those occurring during long-term potentiation. Most importantly, This training blocked the ability of a later experimental manipulation to produce long-term potentiation, presumably because the synapses were already potentiated. Long-term potentiation has been studied most thoroughly in the hippocampus, but it occurs in many other brain regions, as well as the spinal cord. The ubiquitous nature of this phenomenon has led to the proposal that long-term potentiation is a synaptic record of past experiences. This position may be too strong, as synaptic plasticity could occur through more than one mechanism, i.e., long-term potentiation may not be the only one. Nonetheless, although many details remain to be established, most scientists agree that long-term potentiation is a key mechanism by which experience modifies behavior. Chapter Summary: Memory, the cognitive process of acquiring and retaining information for later retrieval, is manifested as experience-dependent modification of behavior. Memory is a fundamental component of all cognition, in most cases facilitating other cognitive processes. The ability to modify behavior based on experience is heritable and provides evolutionary advantages to species who inhabit unpredictable environments. Memory progresses through 3 phases. Encoding converts sensory input to neural signals. Consolidation modifies encoded representations to increase their stability, and retrieval accesses stored information. Memory may be modulated at any one of these stages by factors such as emotional state, pre-existing knowledge, or other ongoing cognitive processing. Given this ongoing processing, it is unlikely that retrieved memories are a snapshot of past events. Working memory is the active process of maintaining and updating information in short-term store. It is measured in both humans and animals by testing the ability to use recently acquired knowledge following a delay. These tests reveal interspecies differences in working memory capacity that are tightly linked to neural complexity in the prefrontal cortex. This has led to the theory that increased working memory is a qualitative shift in the evolution of human cognition. Reference, or long-term, memory may be conceptualized within a multiple memory systems model, which associates the processing of different types of information with particular neural substrates in the mammalian brain. The largest division is between non-declarative and declarative memory, distinguished by conscious awareness in the latter. Non-declarative memory includes modification of a response to a single stimulus presentation, habituation and sensitization, perceptual priming, classical conditioning, and procedural memory. Declarative memory includes knowledge of facts, semantic memory, and personal events, episodic memory. A heated current debate in comparative cognition is whether an animal's ability to remember the what, where, and when of an incident is equivalent to episodic memory in humans. Memory is mediated by cellular and molecular changes, many of which are conserved across evolution. One example is CREB. Activation of this intraneuronal protein plays a critical role in several types of memory across many species. Intracellular changes, such as in CREB, produce synaptic plasticity that alters neuronal communication. So this is about sensitization. These long-lasting effects may be mediated by long-term potentiation, the persistent increase in synaptic strength as a result of high-frequency stimulation. The properties and underlying mechanism of long-term potentiation make it an ideal, although not exclusive, model of memory.